Turns out that when you have these kinds of mutations to this particular part of this protein, it gets stuck on. So it's constantly sending a signal to the nucleus of the cell saying grow, 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 grow. And that obviously is the basis of a type of lung cancer. So that was pretty exciting. It was good to find that, and this is work that we published in 2004. An interesting add-on to this particular study is that some of the patients whose EGFR genes we sequenced had responded very well to a drug called Iressa. It was a new drug at the time. And Iressa almost didn't get FDA approval because it only worked in a very small percentage of lung cancer patients. But in the 10% or so of lung cancer patients that responded well to it, their tumors basically melted away. It's amazing. And that's why it got approval, because the impact in that small subset of patients was very, very powerful. So everybody wondered, what's the difference? Most of those patients that responded well to the drug were non-smokers. Yeah. So you've heard celebrated cases, I don't know if, so this guy, Chris Reeves, the actor, that was, had the spinal cord injury. His wife was a relatively young woman, never smoked cigarettes in her life, got a very aggressive form of non-small cell lung cancer and died. EGFR mutation. And at the time, if they'd have known this, they could have put her on this drug and it might have made a substantial difference. But this is a relatively new finding. So we wondered, what was the basis of these patients? And it turns out that 80% of patients that responded to that drug had mutations in this gene. So it's pretty straightforward. Now when a patient comes into the cancer center, signs of lung cancer, the oncologist takes a family history, smoker, never smoked a day in my life. Okay? Let's do a quick assay to sequence their EGFR gene, mutation, put them on Iressa right away. The other 20% have mutations in genes that perform a similar function to EGFR, right? So bit by bit, we start to take this apart. So if we can look at these lists of genes, that's great. We find things like this. There are these kinds of success stories. But there's still a lot of things that we're missing, because when we look at only a few genes, we're only looking at a very small part of the genome and only a few genes. So we kind of wonder what we were missing by doing this only on kind of known cancer genes. And we also wonder what we were missing by just sort of focusing on the bits of genes that actually encode the protein, because things can happen to other regions of a gene that can inactivate it, and that could be a problem. So we were really kind of bothered by this. What are we not finding? But sequencing was hard, and it was pretty expensive back around 2003, 2004. Well, we knew of some new technology that was coming down the pike in 2005, 2006. And there's a whole lot of new technology that sort of followed. It's all called next-generation DNA sequencing. And the big difference is this. When we used to do DNA sequencing in the 90s and early 21st century, you basically could sequence about 100 little DNA snippets at a time. It would take a few hours to get that data, and you'd put it in the computer, and then you'd run the machine again and again and again. And it'd just take a long time to get through all 3 billion bases that made up the human genome. But these new technologies used an approach that's called massively parallel processing. In other words, we figured out how to do, instead of 100 DNA samples at a time, 1 million DNA samples at a time, or 100 million DNA samples at a time. It's pretty neat. So this is the first of these very powerful technologies to come out. It was invented by a company in the UK called Solexa. And at the time, we got our first machine in. We actually were able to sequence a gigabase, 1 billion bases of DNA in about a week. That was about a 10,000-fold improvement over the technology that we used to sequence the first human genome. And as of last year, this technology, which is now owned by a company called Illumina, based in San Diego, that number had gone up 50-fold over that 4-year period. You could sequence 4 billion bases a week on one machine, and this is one of the Illumina machines. It's pretty cool. It's all sort of compartmentalized in this thing called a flow cell, where you can see how you can connect tubes to either end. All of your millions of DNA molecules are encompassed in this flow cell, and basically the reagents that you need for sequencing flow from one end to the other end, and the sequencing takes place there. This is in reality about the size of a microscope slide, and so it's essentially like a lab on a chip. What happens are you have individual DNA molecules that stick to a small adapter DNA molecule on the surface of that flow cell. We use a type of enzymatic amplification to take one molecule and turn it into these little forests or clusters of molecules. So each one of these spots is about 1,000 copies of the original molecule. And we need to do that so when we shine a laser over these at each cycle, we get enough signal off each spot to basically see color. And there are 4 different bases in DNA, A, C, G, and T. And essentially what we do is we build a fluorescent dye-labeled copy of the DNA that's stuck down to the piece of glass at any one spot. And each one of the new bases that we add is a different color because it's got a little fluorescent tag on it. So at each cycle we can shine a laser across that entire flow cell, and we light up all the different color spots. And depending on if we see an orange flash, it's a C. If we see a blue flash, it's a T. If we see a purple flash, it's an A, and so forth. So you just essentially look at the same area on that flow cell at different cycles, and you see the color change from cycle to cycle, and that's the sequence of the DNA for each one of those spots. So this is a very simplified version. Remember that on that flow cell, we're sequencing hundreds of millions of spots. So tremendous amount of data flows through to that wonderfully efficient, energy efficient data center. And all of this is collected and then analyzed. So this was cool. This was great. And we basically were the first people to say, hey, I bet we can use this for our cancer studies. I bet instead of just sequencing a few hundred genes that we know are important in cancer, we can sequence the whole, well, we can sequence all of the genes. We can sequence the whole genome. And we can find the difference. And what we hypothesized was, you know, the way to do it is this. Let's take some DNA from a patient's tumor, and let's take some DNA from the same patient from some normal tissue, right? So for example, in the case of lung cancer, a little DNA from the tumor that was resected by the surgeon, and a little DNA from some blood cells. And we sequence both genomes, tumor and normal, and we compare them, and we look for the differences that are present in the tumor. The idea is those differences, those mutations are going to be likely important in understanding what went wrong in the normal genome to cause the tumor. So this slide just shows you a representation of one of the first cancer genomes that we sequenced. This is from a patient who has AML, or acute myeloid leukemia. And you're looking in this rather simple looking figure here at their entire genome sequence, both tumor and normal, compared to each other. So here are the chromosomes, chromosome 1, 2, 3, 4, 5, on around to X and Y. This is a female, no Y chromosome data there. And then outside the circle, these little markers show you the locations of the mutations that we found. There's not very many. There's not very many that are different from normal to tumor. But those are the events that likely were at the root of this lady's leukemia. And so we've gone on now and done this for several hundred patients in an effort to learn what are the common changes and how can we learn more about the disease. The technology has gotten even better. This is the current machine from Illumina. Right now, you remember that I told you last year with the Illumina sequencer we could get about 50 billion bases per run. It's gone up another 5-fold since then, and we've actually done experiments on a new configuration that more than triples that number as well. The key thing here is that this all makes the sequencing process cheaper. I'll tell you about in a minute about how expensive it used to be to sequence a genome in the early days with the old technology. But right now, we can take both tumor and normal genomes, sequence them completely, do all of the analysis for about $40,000 per patient. It's still expensive, but we've come a long way. So we really can do experiments like this. We really can take 50 people with leukemia, 50 people with lung cancer, sequence their genomes, both tumor and normal, and start to dig down at the molecular changes that underlie their cancer. This is the comparative slide. So if you had come to me 6 or 7 years ago and said, hey, I'd like you to sequence my genome, right, and the technology that we had back then, you would have had to written me a check for 15 million bucks. Now, if you come next week, we'll have to talk about a lot of other things, so you can't just write me a check for 15,000. But if you could, that's what it would cost. That's actually the cost of sequencing a single human genome, right, with a minimal analysis, $15,000. So 3 orders of magnitude decrease in cost over a 7-year period, and this is going to go down even further in the next couple of years. So it really allows us to do some powerful things in cancer, but as well as in some other diseases that we're just getting underway. Okay, so that's sort of the background. That's kind of the technology and its the application to cancer in general. I want to spend the rest of my time telling you about some stories, some sort of things that we've done with this technology, and I want to focus them on one disease, which is AML. So this is a disorder of hematopoietic stem cells. Basically, you start with stem cells, and when they acquire these mutations that I'm going to tell you about in their genomes, they lose the ability to differentiate into the mature cell types that you need in your blood system, in your immune system, and so forth. They accumulate in the bone marrow, and they crowd out all the normal cells, and basically you don't have enough white blood cells, red blood cells circulating in your veins and arteries to keep you alive. And it's a pretty awful disease. And this is just showing, this is a zoomed in version from right here in this bone marrow sample where the marrow is just clogged up with all of these myeloblasts that basically don't do anything except take up space and burn energy. So this is what happens in your bone marrow and then out to your bloodstream. You have these hematopoietic stem cells. Their job is to turn into things like monocytes and red blood cells and megakaryocytes, the B cells that make antibodies and the T cells that kill virus and bacteria and so forth as part of your immune system. What happens is that AML essentially arrests differentiation of these early precursor cells at various times, and you can see M1, M0, M3, and stuff like that. These are just all of the different subtypes of AML, and the classification is based on where differentiation gets hung up. Well, in terms of the genomics of AML, especially when we started, very little was known about the initiating mutations that cause the disease, and we wanted to try to dig down and get some idea of that. Most of the, if you take these bone marrow biopsy samples from AML patients and you look at the chromosomes look normal in most patients. In some types of lung cancer and breast cancer, the chromosomes start to become sort of a tangled mess, and they break apart and they lose sections. You may have 3 of one particular type of chromosome. Part of one chromosome might have broken off and gone to another chromosome. And those are sort of gross mutations that you can actually see under a microscope. But in most of these AML patients, there are no such changes of the chromosomes. They're cytogenetically normal. And so looking at chromosomes doesn't really tell you much.